Welcome back to the Chitin Corner. We're so glad you're here to learn all things fungal. Sit back and relax. This will be fun. Molds, and particularly hyaline molds, comprise a hidden universe that exists all around us, rarely noticed until they become opportunists. Hyaline fungi are often beautiful when grown in culture, presenting with a wide array of colors and shapes. But what do they mean? In patients, they can become involved in anything from allergies to systemic disease and meningitis. This is the second video in a series designed for the field of medical lab science, for students preparing for the ASCP exam or equivalent, or for those already in the field looking to expand or refresh their knowledge. Each organism discussion will be from a laboratory perspective with emphasis on clinical significance and diagnosis. These top 10 hyaline molds presented here barely scrape the surface of the entire hyaline fungus cohort. They were chosen based on their ability to cause disease, their prevalence in medical mycology, and their appearance on board exams. This content should not be taken as medical advice and is not intended to guide diagnosis or treatment. Hyaline molds are those that lack pigment in their structures. This means they have clear or colorless hyphae, conidiophores and conidia in nature. On the microscope, they often appear blue due to a common dye, lactophenol cotton blue. This image on the right is of a dematiaceous fungus. These have a tan, olive, or brown pigment in the hyphae, and typically in the colonies as well. On the plate, hyaline fungi have a white, pale beige or tan color on the backside of the agar plate or culture. When looking at the backside, you're actually looking at the hyphae. The color of the hyphae decides whether a mold falls under the hyaline or dematiaceous group. These two groups account for the majority of molds recovered in clinical cultures of patients. For this video, we will focus on hyaline fungi. For our mold discussions, we'll be welcoming Fuzzy, who will join us today. He gets the warm and fuzzies when talking about molds. As one of the most common mold isolates overall and one of the most common pathogenic molds, Aspergillus fumigatus comes in at number 1. I often think of Aspergillus fumigatus as the Staph aureus of molds. This will make sense if you've had bacteriology. Basically, it's the go-to pathogen for this group. It is the single most common cause of invasive disseminated aspergillosis, allergic aspergillosis, and fungal sinusitis. The colony, pictured here, is usually described as a smoky gray-green, blue-green, or forest green colony. The microscopic exam of filamentous fungi, or molds, is usually performed with a lactophenol tape prep exam. This is the most effective technique for examining the reproductive structures that are typically born on the aerial mycelium at the top of the colony. The cellophane tape attaches easily to structures, such as hyphae and spores, before being transferred to a drop of lactophenol cotton blue on a microscope slide and cover slipped. Applying tape to a mold and then onto a wet drop of dye on a slide presents a certain learning curve. Don't give up. This hurdle can be quickly overcome. It's the microscopic exam that follows where students often hit a roadblock. Translating what you see into terms and then an identification can be quite challenging. To help us over this obstacle, I've found it helpful to match each organism's microscopic to something more familiar, give it personality, and then attach this to the name. We're going to use this approach before breaking down some of the features. At the end, we look for particular hallmarks for each organism that distinguish them from their neighbors. Back to our organism at number 1 on the hyaline list. Aspergillus fumigatus has uniseriate phyalids. In other words, it has one row of cells that produce the conidia in chains. Conidia are asexual spores. If you're having trouble with these terms, please check out our video Mycology 101, How to Speak Fungus, which will be linked here at the top. The hallmark for fumigatus is that the phyalids attach to the upper 2/3 of the vesicle only. The conidia are also columnar, which means they form a column in nature. Sort of looks like someone going to town on their hair with the blow dryer. Aspergillus niger comes in at number 2 on our list. This species is known for its high affinity for the ear, leading to otomycosis or ear infection. They also cause a type of infection called fungus ball, which is usually respiratory, as well as a cause of disseminated disease. The colony is a good trick question. Is it hyaline or dematiaceous? It has a black peppered surface due to dark conidia, but because it has hyaline hyphae, it falls under the hyaline umbrella. Once mature, it becomes a conidia factory producing what seems like quintillions and sextillions of dark round conidia. If these conidia escape the culture, they can lead to horrible problems with cross-contamination of other cultures. For this one, the microscopic is actually pretty simple. Aspergillus niger is a giant, messy, hairy ball of phyalids and conidia. It's actually biseriate, so there are 2 rows of cells, phyalids and medulla, but this is hard to see. Its hallmark is the ball of phyalids that radiate from the vesicle in all directions. Kind of looks like the fruit from a sweet gum tree. Ranking in at number 3, we have Aspergillus flavus, which is known for its aflatoxins. It is the second most common cause of invasive aspergillosis. Its colony, when fully developed, is a beautiful yellow-green chartreuse or olive to gold color. The colony can become quite woolly in texture. Similar in size and appearance to Aspergillus niger on the cotton blue prep, its conidia are lighter in color. It's known for having both uniseriate and biseriate conidiophore heads, or vesicles. Hallmark is a rough to spiny conidiophore shaft. It kind of looks unshaven. Before we move beyond this dominant genus of hyaline molds, we need to talk about Aspergillus terreus, which comes in at number 4 and the last of the big 4 Aspergillus species. Terreus is an agent of allergic bronchopulmonary aspergillosis, and it may also disseminate to other areas of the body. Its colony is a delicious cinnamon brown color, but may be yellowish or sand-colored. Aspergillus terreus is one of the more difficult to speciate at the microscope. The phyalids are long, thin, compact, and biseriate. The conidia in chains are relatively small. The hallmark is the presence of larger sessile conidia attached directly to the hyphae. These are called allurioconidia. Moving beyond the Aspergillus species, we have Fusarium species, which slides in at number 5 on our list. Known as a common cause of fungal eye infection called mycotic keratitis, Fusarium is also involved in other mycoses. Pretty in pink or purple is how the colony develops after starting out as cottony white. As a hyaline mold, you'll see hyaline septate hyphae on the cotton blue, with 2 types of conidia. Larger multi-celled canoe-shaped conidia, which are the macroconidia, and smaller conidia in masses produced on phyalides. These are the microconidia. Hallmark is the presence of canoe-shaped macroconidia, which serves to differentiate the organism from the next on the list. They kind of look like canoes, and they may grow like bunches of bananas. Acremonium is in at number 6. Although it may be a contaminant, acremonium has been reported as an agent of mycetomas, corneal infections, and nail infections, as well as a rare cause of invasive disease. The colony is usually light in color, white or pale pink, similar to Fusarium but very compact. When it comes to the microscopic, acremonium is sometimes acrimonious because it's basically Fusarium minus the cool canoe-shaped macroconidia. So it has long, straight phyalids that emerge at 90-degree angles from the main hyphae. Free conidia accumulate in masses at the end. Its hallmark is, well, everything, 90-degree angled phyalids with masses of conidia. It kind of looks like cotton candy on a stick. Moving down the list, we move into more saprophytic fungi, or fungi that are infrequent pathogens, things that are often considered collection contaminants. In other words, they're just along for the ride. Penicillium comes in at number 7 as the most dominant of the saprophytic hyaline molds. It is very common and usually not involved in disease, but has been associated with rare cases of mycotic keratitis and automycosis. It is most famous as the original producer of the antibiotic penicillin, and it's also in blue cheese. Bet you'll think twice about indulging in that again. The colony is usually small, compact, and some variation of green, either blue-green or gray-green. Cotton blue is simply the standard aspergillus structures without the vesicle, so branching chains of metalla and phyalids with chains of conidia at the end. To ensure you're dealing with a penicillium as opposed to one of its many doppelgangers, check for the hallmark, which are short, kind of squatty phyalides arranged in a compact way. It sort of looks like a brush or a skeleton's hand. That's right. You thought you weren't this close to the end, unless you were checking the progress bar at the bottom. Number 8. 9 and 10 are all penicillium-like saprophytic molds. Because they are all common contaminants and all resemble each other, we're going to knock them out all at once. Pecilomyces species, Scopulariopsis species, and Trichoderma species are all common contaminants. Infrequently involved in disease, they may occasionally be implicated as causes of things like nail infection, keratitis, and sinusitis, often in immunocompromised patients. Pecilomyces has a yellow-brown or lilac colony. Scopulariopsis is usually a light brown or tan, and Trichoderma has a colony that resembles a lawn with various green patches. On the cotton blue, Pecilomyces very closely resembles Penicillium, but with elongated phyalids, especially at the tip. It has a more delicate look. Sometimes Pecilomyces structures are splayed out or arch away from the central axis. Since the phyalids are elongated, they may resemble bowling pins. Scopulariopsis, on the other hand, is a more robust-looking Penicillium doppelganger with rough conidia in chains. Scopulariopsis conidia are actually produced on annelids. The difference between annelids and phyalids is mostly just a technicality involving how the conidia are produced. On the scope, annelids and phyalids look very similar. Scopulariopsis conidia may be pointed at the end, resembling a bishop's hat. Last and not least, we have the beautiful grassy-looking Trichoderma. This hyaline mold has the same structures as Penicillium, but the phyalides are at wide 45-degree angles from one another, and the conidia are actually in small clusters as opposed to chains. It kind of resembles briar patches. Congratulations for making it to the end of the hyaline journey. I would encourage you to continue exploring, as there are many more cool hyaline fungi out there, some almost alien. They're all around us, in the air we breathe and soil beneath our feet. I hope to see you next time for the dematiaceous fungi. Many images were provided by the University of Adelaide Mycology. See link in the description. Thank you for visiting, and please leave a like, comment, and subscribe.